All right. Can I have your attention, please? Can I have a seat very quickly? All right, so the lecture of today, it's a very long lectures. So I need you to be focused 100% with me, all right? Because they are related to each other, okay? So our lecture is about structures of hemoglobin and myoglobin and connective tissue. What do you know about hemoglobin or myoglobin? What are they? They carry oxygen, yes.
Give the blood their color.
No. Not hemoglobin that gives color. It carries oxygen to the body.
Yes, correct. What else? It's only that?
No.
You know, only that they carry oxygen? They are found in the blood, yeah. So, hemoglobin, where they carry? From where to where? The oxygen. Okay, and what about the myoglobin?
The reverse.
Huh?
Straightened muscle.
Yes, it's from myoglobin فيها كلمة myo. It means muscles. All right? So from the heart and the muscles. So you have to know that hemoglobin, it has two parts. Okay? So we have the hem and you have the globin. All right? So the globin is the protein side. All right? And the hem, it is the group which is inside your hemoglobin which carry the iron metal. So that's why either for the hemoglobin or the myoglobin, both of them, they are carrying the iron. All right? So that's why we call them metalloprotein. Okay? Because they are carrying metal. All right? Okay. So as you said, their function, it's mostly to carry the oxygen. So the hemoglobin, it is found in the red blood cells. The myoglobin, it is found in the heart and in the muscles, all right, skeletal muscles. The myoglobin, they will carry the oxygens to the muscles. However, the red blood cells, they will carry the oxygens from the lungs to the rest of the organs, okay? All right, so we're gonna start by talking about the myoglobin, structures. So last week we spoke about primary structure, secondary structure, tertiary structure, quaternary structures. Okay, and we said that all your protein, they have a tertiary structures, and the hemoglobin, it has what kind of structure? The hemoglobin. Quaternary structures, correct, okay? Because we said that the hemoglobin, it is composed by 4 subunits. For the myoglobin, it is monomeric. It means it is composed by 1 subunit. So this is the structures, as you can see here. You have here helix, alpha helix, this is the secondary structures, which are connected here by non-helical, you can say, the regions which connect your helix together. So we have 8 helix. All of them, they are right-handed helix, okay? And as you can see here, we have the hem, which carry your molecule of oxygen, okay? So these myoglobins, they have only one side for the oxygens, and they carry only one molecule of oxygens, okay? For the hemoglobin, this is what we said last week or last Monday. We said that it is a tetramer because it is composed by 4 subunits, 2 alpha and 2 beta, and that each of these subunits, they have their own tertiary structures, and these 4 subunits, they will be, you can say, assembled through non-covalent bonds, and they will form a quaternary structures, okay? So as you can see here, this is the 4 subunits, and each subunit, it has the hem, and to which it is attached 1 molecule of iron. All right, so we have 4 heme, 4 iron. It means that your molecule of hemoglobin, it carries how many molecule of oxygen? 4. Okay, myoglobin, it carry 1. Hemoglobin, they carry 4 molecule of oxygens. All right, so there is a specificity for hemoglobin, structural specificity, depending if this hemoglobin attach the oxygen or not. So if your hemoglobin, it is free, it doesn't have any oxygen attached to it, we call it deoxyhemoglobin. Okay, so this is hemoglobin without any oxygen attached to it. So the deoxyhemoglobin, this is the hemoglobin that you know. It has, you know, the 4 subunits, the 2 subunits alpha beta, they have their hydrophobic interaction. We have the ionic and hydrogen biointeraction. This is the normal that we saw. This is the deoxygenated without any oxygen molecule. Now, this molecule, it will bind the oxygen. What will happen? So there is some of these non-covalent bonds, they will be broken. Okay, so your hemoglobin, it passes from what we call deoxygenated to oxygenated hemoglobin. So the deoxygenated hemoglobin, we call it the T form, and the oxygenated one, we call it the R form. Okay, T for taut, R for relaxed. Okay, so the T form or the taut form is the kind of hemoglobin without any oxygen attached to it. However, the R form is the oxygenated, is the hemoglobin carrying the 4 molecule of oxygen. Is it clear? Okay. All right, this is important to talk about it, what we call the oxygen dissociation curve. So this oxygen dissociation curve, it's a kind of presentation of the oxygen saturations in what you call the vertical axis or the y-axis according to the pressure, partial pressure of oxygen, which is here in the horizontal or x-axis. Okay, so we're gonna present here the saturations of your binding sites of oxygen. We said that the myoglobin, they have 1 binding site, right? And the hemoglobin, they have 4 binding sites for the oxygen. Okay, so based on that, when you look to the oxygen dissociation curve for the myoglobin, you see that it has a hyperbolic shape. Okay, it's a hyperbolic shape. Why it has hyperbolic shape? Because at low level or pressure of oxygen, already we reach the saturation. Why? Because we have only 1 binding site. Yani from the beginning, from the first molecule, already all the sites, they are saturated. So we reach the 100% already. Okay. Now for the hemoglobin, we have 4 sites. So it will take more time. Okay. So here, as you can see, the oxygen dissociation curve, it doesn't have hyperbolic shape, but it has what we call S shape or sigmoidal shape. It's the S. Okay. Why? Because here at the low level of partial pressure of oxygen, the first molecule of oxygen will bind to your binding site, one of them. Okay. So this first molecule, it will help the other molecule of oxygen to bind the remaining 3 sites. Okay, so by binding the molecule of this first site, we said that they will increase the affinity for the oxygen, the binding for the oxygen, it will increase. So this is what we call the process of cooperativity. You know what does it mean, cooperativity? No. Okay, though you don't know? What does it mean, cooperativity in general? I'm not allowed to say it in Arabic. So this is what one help the other one. Okay, so one molecules of oxygen, it will help the other one, all right, to bind the molecule of hemoglobin. Okay, so this is a kind of cooperations between, you know, the molecules in order to bind the molecule of hemoglobin. Okay, clear? All right, so for the myoglobin, so we have what we said, the hyperbolic. For the hemoglobin, we have the S shape. So I need, from the first one, I'm gonna have a saturation or the fixation or binding of the first molecule of oxygen, and then when I increase, you know, the concentration or the saturation of oxygen, so we're going to have more and more molecule of oxygen. They will bind to their side, and after, when we reach the 4 sites that will be occupied, we reach the plateau, all right, the saturations, the 4 sites, so we have the plateau at the end. All right, so this binding of the oxygens, which is represented here by the oxygen dissociation curve, it can be affected by what we call allosteric effector. So this allosteric effector, what does it mean? Do you know enzyme? All right. The enzyme, you know that they have an active site where the ligand, it will bind, right? So the allosteric site, it's another site which is beside the active site. So imagine you have your enzyme, I have active sites, and beside it, I have what we call allosteric site. Okay, this is another site where other molecules, they can bind and they can affect the activity of my enzyme. Okay, it doesn't touch the active site, it's another one. Okay, it's independent site. So the allosteric effectors, they are kind of molecules, okay, which can bind to the allosteric sites, and they will change the binding of your oxygen to the hemoglobin. Okay, so this active or this allosteric effector are the pH PCO2. This is PCO2, this is the CO2, the carbon dioxide, which will affect the pH. And we have the other molecule, which is the 2,3-bphosphoglycerate. So we're gonna study both of them, all right? So here, myoglobin, is it influenced by allosteric effect?
No.
It is said no, but you have to tell me why. Why is not affected?
Have only one bond.
Yes, because we have only one site. Allosteric effect is the binding is affected. When you have hemoglobin, we said one molecule will help the 3 other. All right? So I have 4 sites to be filled with the oxygen. However, myoglobin, I have only one site. I have only, so from the beginning, it's already saturated. Okay? The other one, the binding of the 3, it will depends of my effectors. Okay? So this is why for myoglobin, I don't have, so the oxygen dissociation curve, it is not affected by the allosteric effector. However, for the hemoglobin, the binding of the oxygen to the, hemoglobin, it is affected by the allosteric effector. All right. So we're going to study how this CO2, they will affect the pH, how the pH, it will affect your ODC, and we're going to study as well the 2,3-bphosphoglycerate. All right. So let's start by the pH. So the effect of pH on the binding of the oxygen to the hemoglobin, it is called the Bohr effect. It is the name of the scientist who discovered this. اسمه Bohr. All right. So we call it this effect the Bohr effect. All right. So as you can see, this is the oxygen dissociation curve of the hemoglobin. All right. So when you look here to the curve, when you have a low pH, for example here 6.8. So what I have, when you look here to the pressure, and the saturation of oxygen, you see that if I am increasing the saturation, if I'm increasing the percentage of oxygen, okay, and we can say the curve, it can take time, you know, to, you can say, to increase, all right? So it means there is less affinity. The oxygen doesn't have a lot of affinity to the hemoglobins, okay? So it takes times till the oxygen, it will bind to your molecules. So I need really to increase the concentrations of my oxygen until this oxygen, it will bind to my hemoglobin, all right? So at low pH, you can see here that I take more time and I take more oxygen here to saturate my binding sites. If I increase the pH, it will be less. So I will need less oxygen to saturate it, and so on. So when I have low pH, so the affinity means of the oxygen to my molecule of hemoglobin, it will be low. So my ODC, my curve, it is shifting to the right. It moves to the right. You see it here. It starts, normally it has to be here, but it shifts, it becomes in here. Why? Because I need more concentrations of oxygen, okay? So if I need more oxygen here, so which form I will promote, the T or the R? The T, it's the deoxygenated. Will R, it's the oxygenated. So which one no, it's the T. It's the T who it is promoted. Why? Because these molecules, we can say of hemoglobin, they don't have affinity. They don't bind very well the oxygen, okay? So they need a lot of oxygen until this molecule of oxygen, it will come and bind to it. So the form which is promoted is the one which doesn't have oxygen. It's the deoxygenated. It's not the one which have oxygen, okay? You understand? So the opposite conditions is when I will increase the pH, so I'm gonna have the reverse, okay? So the ODC, relax. So if I have high pH, so I'm gonna have high affinity. So from low concentration of oxygen, I'm gonna directly have the saturations, okay? So that's why my ODC now, when I increase the pH here, I'm gonna have a shift to the left, movement of the ODC to the left. And here, which form I'm promoting, T or R? Is the R. Okay, so this is how the pH, you know, it can change the ODC, all right, the oxygen dissociation curve. All right. So now we're going to talk about CO2, how the CO2 will affect the pH, and consequently we'll have this kind of, we can say, figure of the ODC. So, can I have your attention for this one? CO2, usually it is released from the tissue, from the cells. And the one who exchange it, it will be exchanged with the oxygen by the RBC, the red blood cells. The red blood cells, they give tissue oxygen and they will take the CO2. All right. So this is the process. We want to know what's happening, how this CO2 is taken and how the oxygen is delivered. So CO2 here, this is in the tissue, comes the RBC. The RBC, it will take the CO2. Inside the CO2, your CO2, it will combine with the water inside the RBCs, and they'll form for us what we call the carbonic acids, the H2CO3, okay? The H2CO3, it will dissociate spontaneously. Once it is formed, it will dissociate. It will give me H+, which is the proton, and it gives me bicarbonate. Bicarbonate, it's a buffer of the blood. You keep it in mind. is the one who can adjust the pH of the blood. Now if I have a proton, if the proton increase, the pH, how it will be? Decrease. So if I go here back, I'm going to have a pH decrease here. So I'm going to have a shift to the right or left? pH decrease. I'm going to have right. I have here decrease of pH. So I have here increase of proton. If I have increase of proton, it means my pH decrease. If my pH decrease, I'm going to have a shift to the excellent. All right. So now what happened? In the RBCs, I have my hemoglobin, which is carrying the oxygen. Okay. Now, what will happen in the presence of this proton? We're going to have exchange. The oxygen, it will be released and it will be replaced by the proton. Okay. So I'm going to have release of oxygen and I'm going to have HbH, hemoglobin H. And instead of having oxygen, I'm going to have the proton attached to it. Okay. And then the oxygen after, it will be delivered to the tissue. So this is how the oxygen, it is exchanged with the CO2 between the tissue and the RBC. Okay. It takes CO2, combine with water, it's become carbonic acid. This carbonic acid, it will dissociate, it will be cleaved. It will give me proton and bicarbonate. So this proton, it will be exchanged with the hemoglobin oxygen here, and it will release my oxygen, which will be delivered to the tissue. So here you saw how at the level of the tissue, I'm going to have a pH decrease. Okay, so I'm going to have a shift to the right. Okay. Now, things are different at the level of the lungs. Okay? لأنه the lungs, what we're going to have? I'm going to have high concentration of oxygen and low concentration of CO2. So it's like the opposite condition in the tissue. Okay? So I'm going to have low concentration of CO2. It means I'm going to have low H plus, low H plus, high pH. So I'm going to have a shift to the left. Okay. So this is to show you how pH and CO2, they are linked. Okay. And how the CO2 is an allosteric effector, how it can change my ODC. Okay. How it can change, the shifting, of the oxygen dissociation curve. My second allosteric effector, it is called 2,3-bphosphoglycerate. 2,3-bphosphoglycerate is a molecule, is a metabolic molecule which is released by the erythrocytes, by the red blood cells during the degradation of glucose. Okay, when the glucose it is degraded in the RBC, they release a molecule which is called 2,3-BPG. Okay, so this 2,3-BPG, it has, you can say, affinity for the binding site of the oxygen. Okay, so what we'll do, the BPG, it will bind to the active sites of the hemoglobin. Okay, instead of the oxygen. So what kind of hemoglobin will be promoted here? T or R? It's a T. Because the oxygen, it will not bind. Okay? So I'm going to promote the T form. The deoxygenated form will be promoted. So I'm going to have shift to the right or to the left? To the right. Excellent. Okay? Okay. All right. So human hemoglobin, you know that we're going to have in our body, we have kind of two types, not really three types, but we need to mention it. The most abundant form that we have in our blood, it is called hemoglobin A. Sometimes they call it hemoglobin A0. Okay, so it represents like 90% of the total, of hemoglobin. And the compositions is the one that we know. It's alpha 2, beta 2, the 4 subunits. The other form, which is less, we can say, present, we can say, in terms of percentage, we call it HbA2. The HbA2, it is present like by 2 to 5% in our blood, okay? And this HbA2, it is composed by 2 alpha and 2 delta. It's not beta, delta, okay? 2 delta chain, all right? And then we have another type of hemoglobin. We call it hemoglobin F, or we call it fetal hemoglobin. So from the name, it's fetal. It means that his hemoglobin, we find it in the fetus, okay? So when someone, when a lady is pregnant, the most of the hemoglobin that has this fetus, it's hemoglobin F, okay? It's like, it doesn't have hemoglobin A, it has hemoglobin F, okay? So this hemoglobin F, after birth, it will decrease and it will be replaced by hemoglobin A, okay? So in adults, we have hemoglobin A. In fetus, and until we can say 6 months after birth, so we're gonna have this hemoglobin F, okay? So the hemoglobin F, it will represent maximum 2%. We can't find it, okay? It's only 2%, but in adults, most of our hemoglobin is hemoglobin A, okay? All right, this is another kind of hemoglobin, which is called hemoglobin AC, okay? So this hemoglobin A1C, which is, we call it glycohemoglobin, it is used in the diagnosis in the lab. So any doctor, he can prescribe it in the lab analysis, and this to check if this patient is diabetic or pre-diabetic or not at all, okay? He is healthy. So this HbA1c, it is composed, it's the normal hemoglobin, but it is attached to it glucose, okay? So as much as you are eating glucose, as much as glucose, it will bind to your hemoglobin, all right? It will be attached to it. So we have, all of us, we can say we have hemoglobin A1c, but in the kind of a range which is, it has to be less than 7%, okay? Usually the range for hemoglobin 1c, it's, we can say it maximum 5.7%. People who they have, for example, between 5.7 to 6.9, we call them pre-diabetic. Yani they can be predisposed. They can, if they will maintain their lifestyle like that, they will become diabetic. We call them pre-diabetic. And people who they have more than 7%, they are diabetic. Okay, so this kind of analysis, so it is used by the endocrinologist or any doctor to check if your patient it is diabetic or not. Okay, because this HbA1c, it gives us a measure of the level of your glucose 3 months ago. Okay, why 3 months ago? Because the lifespan of RBC is 3 months, is 120 days. So I can have the history of my patient 3 months ago. Okay, I know that 3 months ago he developed diabetes. Okay, you have question? What A or B? لا لسه ما وصلنا لك. This is related to insulin body. The measurement of insulin. Type 1 or type 2 is different. Okay? Type 1 doesn't have insulin. Type 2, they are insulin resistance. It's different. Okay? All right. So this is what you have to know about hemoglobin. So generally, if I summarize, please, can I have your attention and stop talking, please? So hemoglobin, myoglobin, both of them, they carry oxygen. Myoglobin, they carry to the heart and skeletal muscles. Hemoglobin, they carry from the lungs to the rest of the organs. Myoglobin, they are monomeric. Hemoglobin, they are tetrameric. Okay? The hemoglobin, they have T form and they have the R form. The T form without oxygen, the deoxygenated. The R form is the relaxed, and it is the oxygenated form. All right? And the difference between them, that the T, it become R by breaking some bonds, non-covalent bonds, between the alpha and beta dimer. Then we spoke about the ODC, the oxygen dissociation curve, and we said that the myoglobin, it has a hyperbolic shape because we reach directly saturation of the sites of the oxygen in the molecule. However, for the hemoglobin, the ODC is, what is the shape of it? S, is sigmoidal, all right? Because it takes time, because we have 4 sites, so we need more oxygen to fill these 4 sites, okay? And we said that the first molecules of oxygen which will bind to your hemoglobin will help the 3 remaining to bind the rest of the site. This is what we call cooperativity, okay? They cooperate to bind. And then we spoke about the allosteric effector, the molecule which will affect this ODC, okay? So we said when the pH decrease, we're gonna have a shift to decrease, you have the right. If it increase, to the left, okay? And this is related to CO2. And when we have a high CO2, we're gonna have a shift to the right. And when you have at the level of the lung, we're gonna have a shift to the left. All right? Then we spoke about hemoglobin. We have 3 types. Hemoglobin A, hemoglobin A2, and hemoglobin F, okay? F is the one which is at the lowest, less than 2%, and we can say the major hemoglobin types is the hemoglobin A in our blood. All right. Always, there is diseases which are related to hemoglobins, okay? And this is what we call hemoglobinopathy, okay? So this hemoglobinopathy, here in Bahrain, for example, we have a lot of cases of sickle cell diseases, okay? There is a lot of people who are sickler. We have other types of disease which are thalassemia, alpha thalassemia, and you have beta thalassemia as well. So I'm going to just speak about sickle cell disease today. I'm not going to speak about thalassemia. So sickle cell disease, what is, it's due to what? What is the causes of it? The cause of sickle cell disease is just mutations in the beta chain, in the gene which code for the beta chain, okay? We said hemoglobin is composed by alpha and beta, right? So the beta chain, the gene which will give me the beta chain, it is mutated. It has mutations in the 6th codon, okay? In the 6th codon of the genes, I have mutations, so which will lead to when we do a translation, I'm going to replace glutamic acids. It will be replaced by valine, okay? So I'm going to change it from hydrophilic to hydrophobic amino acids by just 1 mutations. So I'm going to have a change of glutamic acids by valine. So by this change, I'm going to obtain what we call hemoglobin S. Okay, it's not anymore hemoglobin A, it's hemoglobin S. And the difference, as you can see, this is the normal hemoglobin. The normal hemoglobin, as you can see, it is round, it's biconcave shape. However, here we can see it is like, we call it S shape or crescent moon, like the crescent of the moon, all right? So it is a sickle cell diseases. Okay. All right, and this sickle cell disease, because they have the valine instead of the glutamic acids, and I said the valine, it is an hydrophobic amino acid. So what will do the hydrophobic? It will escape water. So what they do, this sickle cell, so this sickle cell, they become sticky. They will, aggregate together in order to escape the water. So that's why they will block, the flow of the blood inside the arteries. Okay, so that's why these people, they will have a lot of blood transfusions. They will change for them the blood in order to remove it, and they have a lot of crisis, we call vaso-occlusive crisis, because the blood is not circulating in the vessels. Okay, they will have occlusions. Okay, is it clear?
Yes.
Okay. Now we move to the connective tissue. So connective tissue, it is, from its name, its function is to connect tissue and to support. So structural support and connection, these are the two functions of the connective tissue. The component of the connective tissue, I'm going to have cells and I have protein. So as you can see here in the pictures, I have fibers, protein, collagen fiber. I have elastic fiber. This is the protein fiber which are present in my connective tissue. But I have also different types of cells. I have, for example, stem cells. I have macrophage. I have lymphocyte. I have fibroblast. I have adipocytes or fat cells. So in my connective tissue, I have cells and I have fibers. Okay. So we're going to start first by talking about, I'm not going to talk about cells, we're going to talk about the protein fibers, okay? So the first one is the collagen. Everybody know here about collagen, especially in cosmetics, okay? So because collagen, it is everywhere in our body, okay? In our skin, in our bones, in our teeth, everywhere you have collagen, okay? So it's very important protein fiber. So this collagen, it has a property which is called tensile strength. What does it mean? So it try to tear your skin, okay? You cannot tear it, indefinitely, so that you can't find a kind of resistance. So this resistance is due to the collagen, okay? So that's why the collagen, it gives the strength, okay, what you call it tensile strength, to oppose any tearing, any stretching, okay? In terms of structures, the collagen, it is composed by 3 chains. This is one chain, the pink, the green, and the blue, 3 chains, and they are forming an helix, a triple helix. Okay, we have 3 chains, so they will form a triple helix. Okay, and this helix, of course, to maintain these structures, we need a non-covalent bond, which are the hydrogen bond. This is the bonds which will maintain the structures of your helix. Synthesis of collagen, how our body, they synthesize collagen. So the collagen, they are synthesized by cells which are called fibroblasts. So they synthesize the collagen as a precursor, which will, after it will transform into collagen, okay? So the precursor, it is called procollagen. So the fibroblasts will synthesize procollagen, and then this collagen, it will form what we call the collagen fiber. All right. All right. In terms of structures, primary structures of your collagen, it is composed by a triplet. As you can see here, this is my sequence of amino acids. Okay. So you see here, every time I have a glycine, glycine, glycine. Every 3 amino acids, I have glycine, all right? So the most abundant amino acids in my collagen structure is the glycine, okay? Then, after that, as we said, glycine X and Y. The X, the second one, mostly it's proline, okay? But it can be another types of amino acids. For example, here I have leucine. Here I have alanine, but mostly it's proline. Okay. Then the third, the Y, it is hydroxyproline or hydroxylysine. What does it mean, hydroxyproline or hydroxylysine? Huh? What is lysine? What is proline? Amino acids. When I said hydroxy, they have OH. Okay, they have an extra OH in their structures. Okay, so that's why we call them hydroxyproline or hydroxylysine. Okay, so these hydroxyproline or hydroxylysine, they carry glucose. Okay, they carry a carbohydrate sugar, can be glucose, can be galactose. So that's why your molecule of collagen, it is called a glycoprotein. Okay, so the collagen, we said that is protein fiber. Okay, because الشكل تبعها is fiber. All right, so this protein fiber, it is attached to it sugar, which can be glucose or galactose. So this makes your molecule a glycoprotein. Okay. All right, these are the steps for collagen synthesis. Very quickly, as all the protein, we start by the gene. The gene, then I'm going to have transcription, I'm going to have messenger RNA, and then I'm going to have translation, I'm going to have my protein. So these, my DNA, it will be transcribed into messenger RNA, and then this messenger RNA, as you can see here, we have the ribosomes, all right? They will transcribe, we have my here protein, okay? So this protein, it is called pre-pro alpha chain, okay? We call it pre-pro, okay? So this pre-pro alpha chain, in the N-terminal, the first part, the first amino acids, it has a sequence of amino acids which call it signal peptide, okay? So after, you know, the finishing, we can say, of the translation, this signal peptide will be cleaved, will be removed from the structures, okay? And your protein become called pro-collagen. So it was pre-pro, now it's become pro-collagen, all right? Pre-pro alpha, and then it become pre-pro collagen, all right? So now, you saw with Dr. Ramey or Dr. Osman that during translation, we can undergo also post-translation modifications, right? By addition of OH group, by addition of any components, okay? So, based on the structures, here we have hydroxyproline, hydroxylysine. These are amino acids to which we added OH. So, they undergo post-translation modifications, okay? So, these molecules, as you can see here, we add to them OH. Okay, so this is the post-translation modifications, okay? So I have here a first chain, then I'm going to synthesize a second chain, then I synthesize a third chain, and after that, 3 of this chain, they will combine together to form my triple helix, okay? Because the collagen, it is composed by 3 chains, right? And they will compose and they will arrange in what we call a triple helix. So once it is formed, this triple helix, it will be released in the extracellular matrix, okay? In the extracellular matrix, this we can set pro-collagen, all right? We're going to have a cleavage of the C-terminal. I'm going to have a cleavage of the N-terminal here. And then I'm going to form what we call the tropocollagen, which is my collagen fiber, all right? And then after this, this is my collagen fiber. This is one. This is the second one, which is released. This is the third one, which is released. And these one, they will form, as you can see here, a cross link, okay? They will make a cross link between each other. This is what will make the collagen very, tensile strength very great. Okay, so they are cross-linking between the different here chain of the collagen. Okay. Diseases related to collagen deficiency, we call them collagenopathy. There is an example which is called Ehlers-Danlos syndromes. So this kind, you can look at it, it's not really. So they are, characteristic by the skin extensibility. You said that when you try to extend your skin, you cannot. There is a limit, all right? So this kind of people, they can extend it interminably. And they have this kind of joint hypermobility where they can, flex their, their joint more than normal. The second diseases, which is called osteogenesis imperfecta. So this kind of people, they have kind of this twisted spine. But the most important, features that the sclera, the white of the eyes, it's become blue. All right. So it's kind of very, you can say, features. So when you look to the eyes, so the white, it's blue. All right. So this is a kind, it can give you an idea that this person, it has osteogenesis imperfecta. Okay? All right, let's finish the elastic fiber. So elastic fiber, it is composed by two types of proteins. 90% is elastin, and the other remaining, the 10% is fibrillin. Okay, so it is composed by only these two. So the elastin fiber, very briefly, it is responsible for the elasticity of tissue. So from the name elastic, okay? So we can find it in the tissue which require this kind of elasticity, like the lungs, like the uterus for pregnancy, the vessels, etc. So all these tissue, they are very rich in elastin, okay? Same thing. For the elastin, it is synthesized by the fibroblasts, same like the collagen. Collagen, it is synthesized by the fibroblasts. As well, the elastin, it is synthesized by the fibroblasts. And also, they are synthesized as a precursor. Collagen, it is synthesized as tropocollagen. Elastin, it is synthesized as tropoelastin. Okay? So it's kind of similarity here, all right? And then after, they will do, as you can see here, this is my tropoelastin molecule, what you can see here in blue. And same like the collagen, we're going to have like a cross-link, which will make the elastin fibers, okay? So the elastin fiber, normally, in terms of structure, we said that the collagen, what kind of structure it has? Triple helix. Elastin doesn't have any structure. It's just a random coil. Okay? So that's a kind of coil. It's like, imagine you have the cord of the phone. You remember the old phone where they have a kind of cord or right? You remember it? So when it's not very well, stretched, so it will recoil again on each other. So yes. So this kind of elastin doesn't have any specific tertiary structures, so it's a kind of a random coil. But when there is, stretching, so they can stretch. And after, when there is, relaxing or something, so they will come back to their state. Okay, so they can stretch, but after that, they can recoil again. Okay. All right, this is important by saying that this elastin, it can be decreased in our body if you have an overproduction of an enzyme which is called elastase. So from its name, elastase, it will degrade my elastin. Okay? And in our body, to avoid this kind of overprotection, our body protects us by producing what we call alpha-1 antitrypsin. Alpha-1 antitrypsin, it will inhibit the elastase. So it will prevent this kind of, damage in the elasticity in our tissue. However, there is diseases which is called emphysema, or we call it COPD, chronic pulmonary disease, COPD, pulmonary obstructive pulmonary disease. So these people, which they have emphysema, they have really, we can say obstruction in their airflow. They breathe very, very, difficulty they have in breathing. And this is due by what? They don't have alpha-1 antitrypsin. So they have mostly elastase, which will degrade the elastin. So we don't have anymore this kind of elasticity in the lungs. So people, they cannot breathe very well. They have this kind of difficulty. Okay? The second and the last one, they call it the Marfan syndrome. The Marfan syndrome, it's a disease which is due to mutation in the fibrillin gene, the gene which will produce the fibrillin, all right? So we have this kind of syndromes. As you can see, this kind of people, they are very tall. They have long arms and legs, fingers, and even toes. They have very long toes, okay? All right, thank you. This was a long lectures, right? Yeah. All right. All right, so let's start our lecture. It's very long lectures, so it will be a bit fast. So that's why I want you to be focused 100% with me, okay? All right, so today we're going to talk about structures and functions of myoglobin, hemoglobin, and connective tissue. So what do you know about hemoglobin, myoglobin?
Hemoglobin, it's in the blood.
Okay, it's in the red blood cells. What else? They carry the oxygen. Yes, very good. What else? Myoglobin are nerves. They are what? In nerves? What do you mean in nerves? Ah, no, this is myelin. It's not myoglobin. Myoglobin is mostly in the heart and in the muscles. So from its name, myo, it's muscles. Okay? Yes. Yes. So, hemoglobin, they are composed, we can say, by two parts from its name, hem and globin. Globin, this is the protein part, okay? The hem is just a group which is carried by the hemoglobin, and this group, it is called prosthetic group. It means that we cannot remove it. It is there, it cannot be removed. And this hem, it carry a metal, which is the iron, okay? So that's why this hemoglobin, it is called metal protein, because they are carrying the iron, okay, a metal. So we call them metal protein. Same thing for the myoglobin. Myoglobin also, they are metal proteins, okay? So, as you can see here, myoglobin, it is a monomeric structure, right? They have monomeric structure. However, hemoglobin, they have what kind of structure have the hemoglobin? You remember Monday we spoke about protein structure, right? And we took the example for hemoglobin. Quaternary structure. So we said all the protein, they have tertiary structures, and some of them they have quaternary structures, okay? And one of these protein molecules are the hemoglobin. So myoglobin, as we said, it is monomeric, and if you look to the tertiary structures of this myoglobin, you can see it, it is formed by helix, okay? It is a right-handed helix, and it is connected here by a we can say helical regions, okay? Doesn't have any structure, it connects the helix between each other. So I'm gonna have 8 helix in the structure, in the tertiary structures of my myoglobin. And as you can see here, I have also a hem. This is the prosthetic group to which it is attached my O2, oxygen, okay? So this is the atom of oxygen, it is attached to it. Now for the hemoglobin, we said it is a tetramer. It is composed by 4 subunits, and we have a quaternary structure for it. And these quaternary structures, they are held by non-covalent bond. You remember? Very good. So these are the 4 subunits, 2 alpha and 2 beta. So I have here alpha beta 2 dimer, all right, linked by hydrogen bond and by ionic bond, and each of these, you know, chain, they have their own tertiary structures, and they are assembled into quaternary structure, and each of them they have heme, and each of these heme they carry the iron. Okay. All right. For the hemoglobin, there is something what we call the T form and the R form. So what is the difference between T form and R form of hemoglobin? So the hemoglobin that we spoke about it on Monday, it is the hemoglobin which doesn't carry any oxygen molecule. Okay. So these molecule, we call it deoxyhemoglobin. Doesn't have oxygen. Deoxyhemoglobin, and it is what we call the T form, or we call it the TOT form. Okay, so the TOT form is the form of hemoglobin which doesn't carry oxygen. Okay, now if the 1 molecule of oxygen or 4 molecule of oxygen, they will bind to your hemoglobin, so there will be, as you can see here, some of the ionic bonds that it has, the hemoglobin, will be broken. Okay, so your hemoglobin, it will change kind of structure. All right, so and it will take what we call the R form, or the relaxed form, or we call it the oxygenated form. Okay, so the R form is the oxygenated form of the hemoglobin, is the hemoglobin which is carrying the oxygen. Okay, clear? All right. Oxygen dissociation curve. This is a kind of presentations of the oxygen saturation. So you know that I have a site for oxygen binding, right? How many I have in myoglobin? How many binding site of oxygen I have in myoglobin? In myoglobin, how many binding site I have? 1. In hemoglobin, I have 4. So this is the oxygen saturation. It means how many sites have been saturated by the oxygen. Okay, and in the horizontal line, or x-axis, I have the partial pressure of oxygen. This is the pressure of oxygen, so I'm increasing here the oxygen pressure. Okay, and here I'm looking how many sites have been saturated, or how many sites have been occupied by the oxygen. Okay, so when you look to the oxygen dissociation curve for myoglobin, it has a hyperbolic shape. Why it has hyperbolic shape? Keep in mind, I have one site for myoglobin, right? One site for oxygen binding. So from very low oxygen level, we can say, already my site is already saturated, because I have only one site. So I reach very quickly my plateau, my saturations. Okay. However, here for the hemoglobin, I have 4 sites to fill. Okay, 4 sites of binding of oxygen in my hemoglobin to fill. So I need more oxygen, right? It's not only one. So that's why the ODC for hemoglobin, it has what we call an S shape, or sigmoidal shape. So here, from the first one, I'm going to have, for example, here, the binding of my first molecules. Then this molecule, when it will bind the first site, what will help? It will help the other molecule to bind the 3 remaining sites. Okay? The first one will help the 3 to bind the remaining site. Okay? So when I said that, it means there is what we call cooperativity. Okay? So the first molecule, when it will bind to my binding site, it will increase the affinity, it will increase the binding, okay, of other molecules to the hemoglobin. Okay, so that's why we call it a cooperativity. So that's why at the beginning the curve, we can say it moves very slowly, and then after that we're gonna have this very quickly we reach the saturations. So this is the cooperativity, which means that we're gonna have this kind of cooperativity effect, which increase my affinity to the hemoglobin. Okay, is it clear? All right. So this binding of the oxygen to the site, to the hemoglobin, it can be affected by what we call the allosteric effectors. Okay. Do you know what is an enzyme? Do you know that in an enzyme I have an active site? Okay. So beside this active site, next to it, I'm going to have another site which is called allosteric site. Okay. So in this site, I can bind any other molecules. Okay. And this molecule can affect the activity of my enzyme. Okay. I have my active site where the ligand will bind, but sometimes I'm going to have some effectors, we call them allosteric effector, which can bind this enzyme and can change the activity of my enzyme. Okay. Here for the hemoglobin, I'm going to have some effectors which will change the binding of oxygen to the hemoglobin. So these effectors are the CO2 and the 2-phospho-biphosphoglutariate, the 2,3-BBG. Okay, so we're gonna see them one by one, how they can change, we can say, the saturations, the affinity of oxygen to the hemoglobin. Here I said, is myoglobin influenced by allosteric effect? And I said no. Why?
Because it has only one side.
Yes, correct. Because we have only one side. There is no cooperativity in the myoglobin. And from the first molecule of oxygen, I'm gonna have directly saturations. Okay. The other one, if I will have the first one, I have allosteric, it can change. All right. So this is why the myoglobin is not affected by the allosteric effector. All right. So let's see the pH. So this is my ODC. Okay, this is the myoglobin. It doesn't change, it's not affected by the allosteric effector. We have just to look to these ODC for the hemoglobin. So when the pH it decreases, okay, when you have a low pH. So when the pH decreases, for example, here I have 6.8, this is the lowest one. So what will happen? My affinity for the hemoglobin, the affinity of the oxygen to the affinity will decrease. If the pH decreases, the affinity of the oxygen to the hemoglobin, it will decrease. So it will not bind very easily to the hemoglobin. Okay? So if this is the case, I'm going to have like the curve, it is moving to the right. Okay, as you can see here. So you can see here there is a shift of my ODC to the right, because here I need more and more oxygen because the affinity it's become very low. Okay, so I need more oxygen, so this kind of oxygen they can bind the hemoglobin. Okay, so that's why the pH when it's low, it will promote the T form of hemoglobin. The T form, it means the oxygenated or the deoxygenated? The T form. The deoxygenated. Okay, so the decrease of pH, it will promote the T form. Okay, because it promotes low affinity. Okay, so I'm gonna have more deoxygenated لأنه we don't have, we said affinity for the binding, yes, of the oxygen. Now in the opposite situations, I have high pH. So look here, when I increase the pH, 7, 7.2, 7.4, 7.6. So how you look where the shift goes? Right or left? It goes to the left. Okay, so 7.6, which is this one, the highest one. So I have a shift here to the left. So when I increase my pH, the affinity for the oxygen, it will increase. So I will need less oxygen to bind the binding site for hemoglobin. Okay, so at this moment I'm gonna promote the R form, the oxygenated form. Okay, so this is two opposite situations. So when I have low pH, is mostly the T form. When I have high pH, is the R form. When I have low pH, I have a shift to the right. When I have high pH, I'm going to have a shift to the left. Okay. As a it's not kind of buffer. The buffer it's not the pH. It's the bicarbonate. The bicarbonate is the buffer of the blood. Okay. So now I'm going to talk about the CO2, how the CO2 can affect the pH. So please, I need to have your attention 100% here. So the CO2, I'm going to show it in here. So at the level of our tissue, so if you go to a cell tissue, so we have inside our tissue, we have CO2, okay? And normally the RBCs, they will exchange CO2. They will take CO2 and they will give us oxygen, right? Okay. So we're going to see how this exchange occurs, okay? So here you have my tissue. I have CO2 here. So the RBC will take this CO2, all right? And it will, we can say, combine with one molecule of water inside the RBCs. So it forms what we call a component, which is called carbonic acid, H2CO3. So this molecule inside your RBC, once it is formed, it will dissociate spontaneously without any enzymes or anything. They will dissociate. They will give me the bicarbonate, which is the buffer for the red blood cells, and it will give me protons. When I said protons, I mean by that pH. So here I'm going to have an increase of proton. It means the pH will decrease. Okay, so I have an increase of proton, so the pH, it will decrease. All right, so what happened here is when I have this proton and I have oxygen inside my RBCs, I'm going to have exchange between the oxygen and the proton here, the H. Okay, so this exchange will lead to the release of oxygen. So the proton, they will take place the oxygen, yes. So the oxygen here will be released and it will be, given to the tissue. So this is how the CO2 is taken, exchange it, transform it and combine it with water. It gives us the carbonic acids, and then this carbonic acids, it will dissociate. It gives me the proton, and they give me the bicarbonate, and this proton, they will exchange with the oxygen, leading to the release of oxygen, and it will be taken by the tissue. Okay, so this is how the CO2 is given, and the oxygen, it is taken from the tissue, from the RBCs. Now, if I will apply this to the ODC, to the oxygen dissociation curve. So if I will have here, as I said, high proton, it means I'm going to have low pH, right? So if I have low pH, yes. So I'm going to have the first conditions. I'm going to have a shift to the right. Okay, so this is how CO2, it can influence the pH and it can change the ODC. Okay, now the condition is different when it comes to to the lungs. In the lungs we have oxygen and less CO2. Okay, it's the opposite of the tissue. All right, the other tissue, they are mostly rich in CO2 and they want oxygen, and the lung, they have oxygen, all right, and they have less CO2. So if they have the opposite condition of the tissue, so what I will have? CO2, if the CO2 is low, it means that the pH, it is high, because what I will have here, so I have less of this, less of this, less of this, and less of this. So less of proton, it means high pH. Okay, so if I have high pH, I'm gonna have a shift to the left. Okay, all right, okay, all right. The 2,3-BPG, this is a molecule, this is a metabolite which is produced by our body and specifically by the red blood cells during the degradation of glucose. So when the glucose, it is degraded by the RBC, it produces this product which is called 2,3-BPG. Okay, so the 2,3-BPG, what it will do, it will just bind, it will take the place of the oxygen. Okay, it will occupy the places, okay, or the site of the oxygen. So what I will have, so I will have all my sites, instead of having oxygen, they will be 2,3-BPG. Okay, the 4 sites, they will be occupied by the 2,3-BPG. So at this time, what I will have in terms of ODC, shift to the right or shift to the left? Shift to the right, because I need really a lot of oxygen to remove this BPG and take the place, okay, the oxygen. You understand? Okay, so that's why I'm gonna have a shift of ODC to the right in order to remove this 2-BPG and replace it by the oxygen. All right, hemoglobin. In our body, we have mostly 2 types, but we have a third type, which is very minor, you know, is present in very minor proportions. So the major hemoglobin that you have in your body, we call it hemoglobin A, or sometimes we call it hemoglobin A0. Okay, so this, it represents 90% of your total hemoglobin. All right, and as you know, it is composed by 2 alpha and 2 beta chain. Okay, the second types of hemoglobin that we have in our body, it is called hemoglobin A2. This hemoglobin A2, it is composed by 2 alpha chain and 2 delta chain. Okay, and it represents only 2 to 5% of our hemoglobin. Okay, then we have a third type of hemoglobin, which is called hemoglobin F, F for fetal. So from its name, this hemoglobin, it's mostly present in the fetus than the adult life. Okay, so when you have a fetus, an embryo, they don't have hemoglobin A, they have hemoglobin F. After birth, this hemoglobin F, it will decrease and it will be replaced by hemoglobin A. Okay, clear?
مو مهم هو من وين مصنوع من اي شيء، بس المهم انه هو شلون يتشبع.
شلون؟
يتشبع.
What do you mean by that?
يعني يكون في اكسجين.
Yes, hemoglobin F, it has a high affinity for the oxygen, more than the hemoglobin. This is how the fetus, it catch, we can say, the oxygen from the blood of the mother, because it has more affinity than the hemoglobin A. Okay. There is another types of hemoglobin which is very important, that some doctor, they ask to do it in the blood analysis, and this is to screen if the patient, it has diabetes or it is pre-diabetic. Okay, so they will ask to do analysis of what we call HbA1c. All right, the HbA1c, it's HbA, it's our hemoglobin, but to H it is attached glucose. All right, So as much as you eat glucose, so the hemoglobin, it will attach this excess of glucose to it, okay, to the hemoglobin. So when we measure the hemoglobin A1C, this one, it will give us, we can say, the history of your, you said, your level of glucose 3 months ago, okay, because your hemoglobin, the lifespan, the duration of their life is 120 days, so it's 3 months. So when you measure this, you can know, you know that 3 months ago, so we have this kind of the level. It's not like now we measure it, you know, we can go back 3 months ago, okay, so you can know. And of course, how you know this person it is diabetic? By measuring its level. Usually the level is less than normally. We have HbA1c must be below 5.6, 5.7%. Between 5.7 to said 6.9, these people, we said they are pre-diabetic. So if you have this kind of reference range, so they will, you said you are pre-diabetic, be careful, you can become diabetic, okay? But if they measure HbA1c and they found it's more than 7%, this patient, it is diabetic, okay? It has diabetes, okay? All right. Diseases, some diseases that are related to hemoglobin, we call them hemoglobinopathy. In Bahrain, there is a lot of what we call the sickle cell disease, sickle cell anemia. And we have another types of hemoglobin, which is thalassemia, okay? So the sickle cell today, this is what we're gonna study very briefly. Sickle cell, it is a genetic disorder, it's inherited disorder. It means if your parents, they are sickler, you will inherit that. You will become sickler as well, okay? So it is due to what? It is due to mutations in the beta chain. You know that hemoglobin, it's composed by alpha and beta, all right? So the gene which will code for your beta chain, it has mutations, okay, in the 6th codon, all right? And this, it leads to the, we can say, the replacement of glutamic acids. It means when you do the translation of the messenger RNA, you will have glutamic acids replaced by valine, okay? So this is the mutations. Due to this mutation, we're gonna replace glutamic acids by valine. Glutamic acids is hydrophilic amino acids. Valine, it's hydrophobic amino acids. So if this red blood cells, they have hydrophobic amino acids, what they will do? They will aggregate, they become sticky in order to avoid the water, okay? So that's why they form a kind, they can block the blood flow inside the vessels. So that's why certain patients, they have a crisis, we call it vaso-occlusive crisis, because the blood flow is not anymore circulating in the blood. Why? Because we have this kind of aggregations due to this mutation. And valine, it is hydrophobic. So the red blood cells, they will aggregate together in order to avoid, you know, the water, we can say. So as you can see here, even the shape of this hemoglobin, we call it hemoglobin S. It's not anymore hemoglobin A. It's hemoglobin S. It has like the shape of crescent of the moon, all right? And it's not anymore a round, big concave shape, okay? So it differs. Clear? All right, so we move to the connective tissue. Connective tissue, from its name, its function is connection and structural, to support other tissue, okay? What is composed of? It is composed by protein fiber, collagen, and elastin. This is the two protein fiber which is present in the collagen, and it contains also different kinds of cells. We have fat cells, adipocytes, we have fibroblasts, we have lymphocytes, we have macrophage, we have stem cells, okay? So this is the components of your connective tissue. So today we're going to focus more on the collagen and the elastic fibers, on the protein side. Okay, collagen. Collagen is the most abundant protein fiber in our body. Collagen, you will find it every part, in every part of our body, in the hair, in the skin, in the bones, in the tissue, in the teeth. Everywhere we have collagen. All right. So it's the most abundant protein fibers in our body. So, and it provides what we call a tensile strength. So imagine to take the skin of your face and try to stretch it. You cannot stretch it indefinitely. All right, there is a kind of resistance. Okay, so this resistance is due to the presence of collagen in your skin. Okay, so this is what we said. It gives a strength. Okay, a tensile strength. It opposed to any tearing forces. Okay. In terms of structure, tertiary structures, it is composed by triple helix. So we have 3 chain, alpha 1, alpha 2, alpha chain. They are combined in a triple helix. And of course, this structure, it is stabilized by non-covalent bonds, specifically hydrogen bonds. Okay. Synthesis of collagen. The collagen, it is synthesized by the cells, which call them fibroblasts. They are the cells which synthesize the fibroblast. And the fibroblasts, they synthesize the collagen as a precursor. It's not directly collagen. We synthesize what we call pro-collagen, and then these pro-collagen, they will cross between each other and they will form the collagen fiber after. Okay. So, primary structure, sequence of amino acids, we have a triplet. So, glycine, lysine, hyper, glycine, pro-hype, glycine, alanine, and hydroxylysine. So you see in every triplet of amino, we have glycine. So the most abundant amino acids in the collagen is glycine, and it's the first one. Then it comes the X. X, it can be any types of amino acids, but is mostly, it is proline. This is the proline, which is the most, prominent, in the structure. But we can find alanine, we can find valine. But in terms of proportion, you find more proline than the rest of the amino acids. The next one, the third one, it's hydroxyproline and hydroxylysine. It's either hydroxyproline or hydroxylysine. So, what does it mean, hydroxyproline and hydroxylysine? Do you know what is it? Do you know what is it?
Means OH.
Yes.
And they are acid, but the other.
Yes. Yes. Correct. Yes. Same thing, which is correct. So we study lysine. We study proline. So when it comes to hydroxyproline, it means it's a proline carrying OH group. A lysine carrying OH group. So that's why we call it hydroxylysine or hydroxyproline. Okay? And these hydroxyproline and hydroxylysine, it attached to it in the structure carbohydrate. It can be glucose, it can be galactose. So these molecules are the site of attachment of carbohydrate. So that's why your collagen, it is called glycoprotein. It's a protein which has glucose, it has carbohydrate. So we call it glycoprotein. Okay? Clear? Yes. All right. Now we're going to see the steps. Very quickly for the steps, I don't want you to focus a lot on it. Collagen, we said, is protein. So we always start for the synthesis of protein from the gene, right? From the DNA. Then you're going to have transcription, we're going to have messenger RNA, then you're going to have translations, I'm going to have protein. So these are the three, we can set steps. So here I have my DNA for my alpha chain for collagen. It will be, we're going to have transcription producing messenger RNA. Then this messenger RNA will be read by the ribosomes, here, the black one. And you see here, these are the protein of collagen, all right? So this protein, it is called pre-pro-alpha chain, okay? Pre-pro-alpha chain. You saw also with Dr. Osman that the protein, once they are produced, they can be modified, right? We can add to it some group. I can add methyl group. I can add hydroxyl group. So this is what happens. So you can see here, I'm going to have this pro-alpha chain. I'm going to add to it OH. So that's why OH, I added to which amino acids? Proline and lysine. Lysine. Okay. So I'm going to add it here. Hydroxylysine and hydroxyproline. Okay. So this is where I'm going to add my OH. So this is a kind of post-translation modifications. Okay. The second post-translation modification that my pre-pro-alpha chain will undergo, in the N-terminal of my protein, I'm going to have a sequence of amino acids which is called signal peptide. Okay, it's a sequence in the N-terminal, at the beginning of your proteins. Okay, so this sequence, once your protein it is synthesized, it will be cleaved. It will be removed. All right. So at that time, your protein it becomes called pro-collagen. Okay. So I have a chain, alpha chain, which doesn't have any peptide signal peptide, and it has OH group, and it can also have some glucose or galactose attached to it. Okay. Now, if I will combine three of this, because the collagen is composed by 3 chains, so I take these 3, I will combine them into triple helix. Okay. So this it will form for me, the pro-collagen, and this pro-collagen it will be excreted outside of the cell. Okay. So when it's excreted outside of the cell, N with C terminal of this pro-collagen will be cleaved. As you can see here, we have these dots. This means I'm going to have a cleavage of these N and the C terminal, the both terminal of the pro-collagen. And then I will obtain what I call the tropocollagen or the collagen, simply. Okay? And then this collagen after, this is my collagen, the second and the third. They will cross, as you can see here, we're going to have a cross linkage, and this is what they will form yes. Okay? Is it clear? I know it's a lot. You will need time to process it, but it's not difficult, okay? And any time, if you have any questions or anything, you can come to my office and you can ask me, okay? You are welcome. Okay. Diseases. Always we need to talk about diseases. If there is a deficiency in collagen, we have diseases which we call them collagenopathy. So I choose 2 examples. The first one is called Ehlers-Danlos syndrome. So the first one, as you can see here, they are characterized by we can say, extensibility of their skin. Okay, I told you try to stretch your skin, you cannot. So these people, because they are deficient in collagen, so they can do that. Even for the joint hypermobility, they can flex their joints very easily, all right, without any resistance. The second types of collagenopathy, which is called osteogenesis imperfecta, and this is, we can see here people, they have like twisted spine. It's like S. It's not and the thing which is very characteristic for this disease is that they have the blue sclera. The white of the eye, it is blue. Okay, so it's really something when you look at it, so you think about it, osteogenesis imperfecta. Okay, plus the spine, which is twisted. The last one is elastic fiber. Elastic fiber, which is the second component of the connective tissue, it is composed 90% of elastin, and the rest is called fibrillin. So this is the two components of the elastic fibers. And you have to know that for the elastin, it is responsible of the elasticity of our tissue, right? So any tissue which needs to be, stretched needs to have elastin. So uterus for pregnancy, uterus is very rich in elastin, so it can stretch and it can hold the fetus. The vessels, okay, they can be vasodilations, okay, vasoconstriction. So this kind of, change in the elasticity. The lungs, inspiration, expiration. So these kind of tissue which required, a kind of elasticity, they are very rich in elastin, okay. So same thing. This elastin, it is produced by fibroblasts, same like the collagen. Collagen is produced by fibroblasts, and elastin is produced by fibroblasts as well. Okay, and they are also produced as precursor. So fibroblasts, they produce procollagen. Fibroblasts, they produce tropoelastin. Tropocollagen, tropoelastin, same thing, okay? Then we said that for the collagen, they're gonna have a cross linkage, and they're gonna have my collagen fibers. Same thing here. You have here, this is your tropoelastin, and then you're gonna have a cross linkage between these two tropoelastin, and we're gonna have the elastin fibers, okay? Now when it comes to diseases, before diseases, in terms of structures, we said that the collagen is a triple helix, right? So this is the 3D structures of the collagen. For the elastin, there is no 3D structures. It's a random coil. Okay, there is no helix, there is no beta sheet, nothing. Okay, it's a random coil, and if this tropoelastin, they are arranged randomly, and then when they can be stretched, okay, they can be aligned, and then in resting state they will recoil. Okay, they will. Here we have a stretch, and here when in resting, in the relaxations, so they will become a random coil. Okay, they align or they can recoil. All right, for diseases, to finish, we have in our body sometimes an overproduction of an enzyme which is called elastase. So this enzyme, it will degrade the elastin. So if it will degrade the elastin, it means your elasticity in the tissue, it will decrease, right? Okay. So to avoid this, the body synthesizes what we call alpha-1 antitrypsin. Alpha-1 antitrypsin, it has opposite effect of elastase. So it will inhibit elastase, so it will not degrade my elastin. Okay? So elastase, it degrades the elastin. While antitrypsin, it will degrade elastase. Okay? So they are opposite here action. There is, however, there is people which are suffering from a disease which is called emphysema, or we call it COPD, chronic obstructive pulmonary diseases. So these kind of people, they are lacking alpha-1 antitrypsin. Okay, so these kind of people, they lack elasticity in their lungs. Okay, so that's why they have problems in the airflow, they have problems in breathing. Okay, because their lungs, they are lacking elasticity due to the degradations by elastase. Okay, is it clear? The last one, this is the last slide. This is another disease, we call it Marfan syndrome. and it is due to a deficiency or mutations in the fibrillin gene, which gives us the fibrillin proteins, okay, which is a part of the elastic fibers. So as you can see, this kind of people, they have very long, we can say, arms, very long legs, and they have very, very long as well toes, okay, and fingers. So this is kind of characteristics, but this is what we can be visible, but it can also affect their eyes, their cardiovascular systems, and so on, okay? That's it. Thank you very much, and